Welcome to the Wild Side. That was Norwegian Wood by P.M. Dawn. And today I'm going to be discussing some more recent scientific research, some of the stuff that's happened in the last week or so since I recorded last week's show, but also a few things from before that because sometimes I just stumble upon stuff on the internet, and even if it's a little bit out of date, I just have to share it because it's really interesting. And the first thing I want to talk about today is in fact quite an interesting thing. Maybe a topic that you didn't think you would hear much about when you turned on the radio today, but it is still kind of a cool thing. And it's all about the bacteria that are found in the stomachs of termites as they go about digesting the wood that they eat. And you might not think that termites actually have stomachs because they're so tiny it probably never even occurred to you, but they do in fact. And just like us, they have a lot of gut bacteria that help them process all of the stuff that they eat. And this is actually particularly important for termites because wood is really hard and very fibrous, and so it's actually quite difficult to digest. And for all animals, actually, cellulose and other kind of woody components of vegetation can be quite hard to break down, and this is particularly true for wood. So in order to help them get through all of this process, termites have several hundred different species of microbes within their guts. But these guys are pretty difficult to study because all of the microbes are living really close together in that tiny little space, and so it's hard to separate them out. So we can kind of get an idea of approximately how many species there are, but we can't really isolate them very easily or study them in much detail and understand how each one of them contributes to the larger overall process of digesting that wood. And that is exactly what has been done in this current study. People have actually been able to isolate something. And what they've isolated is an organism that is responsible for a process called acetogenesis. And acetogenesis happens when organisms are able to use hydrogen to convert carbon dioxide into an organic carbon form, and it's called acetate, and that's why it's called acetogenesis. Now, acetate is nutritious to the termites, and so this is something that's quite important to them, and therefore this whole process is a really important part of their daily lives as they go about eating and getting that stuff made for them. And we've known for a while that acetogenesis occurred, and we knew that some of the microorganisms in the termite gut were responsible for this process, but we didn't know exactly which ones. And that is what has really happened here in this study, is that we've kind of moved forward for the first time in identifying those particular things. And in fact, what has happened is that we found out that acetogenesis is performed by, among other things, symbiotic bacteria that live on the surface of protozoans that live within the termite's gut. And that's one of the things that makes this quite interesting, is that it's not just a matter of a single organism, but a couple of organisms that are living in close proximity to each other and having that really close relationship. So the research on this relied on molecular techniques that focused on the genes that are responsible for acetogenesis. So they didn't start off targeting a bunch of bacteria. They didn't just go get a sample and grow them up and try to pick out each different one. What they did was they had some genes that they knew were responsible for acetogenesis, and they used those genes to kind of go fishing to figure out what organisms had those genes within them and were expressing them in order to perform this function in termites. And in particular, the gene that they were interested in was was a suite of genes that were responsible for formate dehydrogenase, or otherwise known as FDH. And FDH genes encode a protein that's necessary for acetogenesis. So if the scientists could find this gene active in samples from the termite gut, then that suggests that that activity was associated with the creation of these necessary proteins. And so whatever cells were creating that protein to begin with, or that transcription of the FDH genes, those were the ones that are responsible for this activity in termites. So it's kind of piecing together lots of little, individual bits of the puzzle in order to get this big picture. It's using this one little thing as a clue to something else entirely. So what they did was they were able to sample en masse the RNA that was found within termite guts, and they were able to sort through all of that RNA, which could have come from anything and everything, in order to find those specific genes of interest. And once they did that, they used a pretty newly developed technique in order to sequester individual microbes into tiny little compartments and grow them up so that they could then see these things all by themselves and really look at their activity in isolation from the activity of all these other organisms that are also in the guts. So many of the microbes that they found in this way had never before been studied. They were brand new to science. And this enforced isolation really allowed the researchers to learn a lot of new things, actually, about what sorts of bacteria are in there and how many are there, and which ones, as I mentioned earlier, are responsible for acetogenesis. And what they found out was that many of the genes that were associated with this product were being produced by bacteria that are known as spirochetes. And spirochetes kind of look like exactly what they sound like they look like. They have kind of a spiral format. They're very common bacteria. We find them all over the place, not just in guts. And these guys actually had previously been predicted by the researchers to perform acetogenesis. But what's kind of cool is that it's not just these spirochetes that were responsible. So they knew that some of the genes were definitely from those bacteria, but they also could tell that some of the genes were from something else, and they couldn't figure out what that other source was. And this was where the mystery really came up and where the study got interesting. And what they noticed over time was that if they looked at where these genes were, that they were unable to kind of pinpoint and unable to assign to the spirochetes, what they noticed time and again was that they tended to be found in the same portion of the termite gut. And in fact, it was the portion of the gut that contained larger wood chunks and bigger microbes that were kind of responsible for breaking those bigger wood chunks down into smaller bits that then other littler organisms could then continue digesting. So everything seemed to be concentrated in one particular part of the gut, which suggests that maybe there was another organism living in that area, all in one little place, that was doing this acetogenic activity. And in fact, a more detailed analysis of samples from only this site found out that the gene that they were looking at was encoded by a previously unknown and unstudied species from a group of bacteria known as delta proteobacteria. So then the authors followed this up by using a color-coded visualization method that allowed them to basically paint the different cells within the termite gut in order to figure out where these bacteria were located. So they kind of knew the general, section of the gut, that bit with the big chunks in it, but they wanted to specifically within that area figure out, where can they find these bacteria? What portion of the gut are they sitting on? Are they on the wall? Are they floating around? That sort of thing. And so what they did was they painted the cells with the FDH genes, and they painted the cells that had another gene that they knew the delta proteobacteria expressed. And what they were assuming was that if indeed these bacteria were responsible for the FDH genes, then both of those different colors would be found in the same place, because it would be the same cells that were producing them. And indeed that is what they found, and so they confirmed for sure that it was these bacteria that were creating these genes. And what they found was that all the bacteria were living right on the surface of little protozoans that were also within the gut. So they had this symbiotic relationship where they were on another organism that was floating around in the termites. And what was really interesting about this was that they knew that the protozoans were producing hydrogen. So these little bacteria were sitting right on top of the source of hydrogen that they needed in order to take that hydrogen and use it to interact with carbon dioxide to produce acetate. So they went exactly where they needed to go to get that hydrogen in a really easy manner and do that thing that they were doing within the termite gut. So it's very efficient. It couldn't have been, more perfectly planned out. So one of the things that the researchers wrote about in their study was not just, these interesting factual results, but also the fact that they really enjoyed having an initial hypothesis that was proven wrong. They had thought that it was purely the spirochetes that were responsible for acetogenesis, but they found out that this wasn't the case, and that, by being forced to kind of think outside the box and try something brand new, they had to use multiple novel techniques and come up with something completely unexpected, which is kind of the fun part of science, because you often you tend to think that you want to just prove yourself right after you've come up with a theory, but actually it's quite exciting to go out there into the great unknown and come up with something kind of on the fly as you go. And this I thought was kind of a cool thing, because it's not often that you see or hear scientists actually admitting this out loud, that they kind of enjoyed making a mistake and having that element of discovery, and I enjoyed seeing that in a scientific paper. And of course there is also the factual information, which is important and not to be ignored. So they discovered both of these facts about the species, where it's located, what it does within the termite gut. And this was more detailed information than they'd expected to get using those initial techniques that they started off with. And this kind of information might be quite useful in the future because it can tell us a lot about how how wood is broken down, the different bacteria that are involved in that process, and how we maybe can work against that, for example, in our houses or in other wooden structures. So all this stuff does also have some kind of practical bearing. So the study in general had lots of things to contribute. Welcome back to the Wild Side. That, as I'm sure you already know, was Coldplay with the song Clocks. And indeed, the next thing I want to talk about is some news about internal clocks, which are something that all of us have, and it turns out that some of us, maybe even all of us, have multiple internal clocks. And that was the result of a recent couple of studies, actually, on some marine species that were found to have internal clocks that were associated with the moon and also the tides, which of course we know are themselves related to each other. So many terrestrial species, from fungi to animals, all have 24-hour clocks that we call circadian clocks. But many other animals can have different clocks. So these are ones that can be associated with other patterns that are more relevant to their daily lives. So for example, as I said, animals that live right along the tide line there on the coast, they might need to have a tidal clock. Or animals that are out in the middle of the ocean that are in the water but they don't have the tides as much, they might have a lunar clock because they can see, or sense, I should say, that type of light as well. And there may be lots of others in addition to those. And it turns out that some of these animals might have multiple of these clocks working together simultaneously in order to tell them different aspects about time. And that is, in fact, the newest result here that I want to talk about today. So these two other clocks that have been kind of discovered and studied in a bit more detail and found to work not just in isolation but with other clocks within these animals' bodies are the tidal/lunar clocks that help things like coastal and aquatic organisms regulate their activities. And these things were studied in an intertidal crustacean called the speckled sea louse, and these guys look kind of like a cross between a pillbug and a tiny little prawn. And then another species called a marine bristleworm, and that sounds pretty much exactly, it looks pretty much exactly what it sounds like it looks like. So what researchers did is they performed environmental and molecular manipulations of the daily clocks of these organisms. And in the sea louse, they found that when that clock was disrupted, these organisms still had a sense of that 12.4-hour tidal clock, and that clock kept on ticking and allowed the animals to stay on track. And the researchers described this as being a hardwired, robust, and independent mechanism that allowed the animals to kind of stay in time as they normally would, even though their regular circadian clock, so that cue they're getting from the sunlight, was a little bit off. Likewise, a disruption of the bristleworm's circadian clock had absolutely no effect on the animal's intrinsic sense of month. And yes, they can actually tell what month it is. And that, of course, is going to be related to moon activity and what phase the moon is in and how much lighting there is, and that's going to change around the year as well. So they were able to figure out and respond accordingly to the time of the year. Now they also found that there are some interactions between the circadian and the lunar clocks, and this is something I've already kind of alluded to. So basically the length and the strength of these different rhythms are going to impact each other. So if one of them is really off, it will start to impact the other one. But if it's just a little bit off, the other one usually can stay pretty steady. And so this seems to be something that in some cases is maybe a bit of a backup mechanism in case one is off. So you might have some really strange weather conditions that keep the sky completely obscured for quite a long time, and so they aren't getting the signals they should have from the environment to reinforce their sense of time. Or maybe for some reason these animals are submerged for longer than normal because, I don't know, there's a shipwreck and the shipwreck covers up the organisms and so they don't get those signals. So having a bit of flexibility is quite good because the environmental conditions might change and throw off one clock for some reason or another. So it's good to have those backups and to make sure that if one of them is a little bit off, it's not also going to drag the other one down with it and completely mess it up. And what's really amazing is that, we've got two animals here that have multiple independent clocks, and that suggests that we probably have lots of other animals that do this as well. And in fact, even in these animals that we've already studied, there might be even more internal clocks that we haven't found. And so there could be layers upon layers of these things that work together to really help us fine-tune our sense of time and our sense of day and our kind of position throughout the year. And all of this is quite interesting because it does kind of raise these questions about internal clocks that we wouldn't have thought of before. I mean, who would have thought we would have multiple things kind of telling us the same basic type of information? But also it raises questions about the molecular and cellular natures of these different clocks. How exactly is it that they're functioning? And how is it that these things can impact animal behavior? So we know a lot about how circadian rhythms can change around the year and cause us to do different things. So for example, when you've got a change in rhythm that causes a migratory bird to decide it's time to leave an area and go to another one. But how do these other things also impact behavior? How does a change in your lunar clock or a change in the tidal clock tell you to do something different, or does it at all? And so it is going to be really interesting to perform further studies and kind of find out what exactly the repercussions of these different clocks are. And kind of knowing that these other things exist out there may be really important for people who are in medicine trying to figure out how to fix individuals that have kind of sleep disorders, or even people who are narcoleptic. So it can be really interesting in the medicinal sense as well, because the more we understand about the evolution of these different types of mechanisms in our bodies, the more we can understand when things go wrong, as well as when things are going right. Now I'm going to shift gears completely and discuss another study that came out recently that was looking at what life was likely to be like early on in the history of Earth. And of course, this is a question that we've been pondering for a very long time, and it was one of the ones that I mentioned last week when I talked about these 20 big questions remaining in science. So people are really interested in better understanding the situation that we had here on this planet early on, and how that situation allowed life to arise. And then maybe applying that information to other planets and trying to figure out what other places out there might have some life like ours, or even any life at all. So the reason that it's in the news right now, this question about the early Earth, is that scientists have just discovered that oxygen most likely appeared in our atmosphere much longer than we originally thought. And by longer, what I mean is about 700 million years. So that obviously is quite a significant difference, even in the terms of how, of Earth years, which of course we are talking in billions of years. So how do we know this? Well, the researchers were in South Africa collecting soil samples, and these weren't just any old soil samples. They're soil samples that were about 3 billion years old, and these are basically the oldest soils that are known on Earth. Now we do also have some incredibly old soils here in Britain, particularly in Wales, if I'm remembering correctly, and a lot of really important geological work has been done from samples that have been taken around here. But these ones in South Africa are even older than those, and so they allow us to kind of look back in time a little bit. And what you can do is look at the chemicals that are in these soils and get an idea not just of how many of those individual chemicals there were, but also kind of, you can think about the chemical processes that created those things. So if you find evidence of certain byproducts, then you know that a particular process was occurring. And what the researchers found here was that there were very low concentrations of atmospheric oxygen. And this, of course, is something that would have been produced by photosynthesis, and so this is a really important milestone in the history of the Earth and all of the organisms that lived on it. And previous studies had suggested, just by way of comparison here, that oxygen didn't begin accumulating in the atmosphere until about 2.3 billion years ago, during something that was known as the Great Oxygenation Event. So this is quite a bit older, 3 billion years versus 2.3 billion years. So that is a significant difference there that has caused us to revise our estimate of when oxygen was produced by photosynthesis. And this suggests that potentially organisms were around for earlier than we thought they were, or that aerobic organisms were around earlier than we thought they were. So we don't know exactly what happened. We don't know exactly what conditions were required for these types of life to emerge, or for certain steps in those life, So for organisms to be able to be anaerobic versus aerobic, we don't know exactly when that shift occurred, although we can estimate. And this new date of oxygen production and possibly consumption makes us completely revise these estimates and maybe move them back quite a bit. So photosynthesis probably did begin earlier than we thought, which means that aerobic life probably emerged earlier as well. And this changes our view of how long it would take certain things to evolve, and it also changes our view of maybe how long a planet needs to kind of get processes going in order to make that planet habitable for certain types of life, like the aerobic life that eventually gave rise to us. So what's interesting is that despite this change in time frame, it's still clear that it can take quite a long time for geological and biological processes to facilitate the sorts of conditions that are necessary to support aerobic life. So even though there is this difference of 0.7 billion years, which again is nothing to sniff at, still it's taking billions of years for life to get to the stage where it can emerge from whatever primordial soup was out there. So that is still quite a substantial amount of time, of course. And this new information maybe puts us one step closer to figuring out how exactly that life did come into being and what conditions were needed for that life here and therefore potentially on other planets as well. We may also be able to use this information to kind of project forward and try to get an idea of what will start to happen on this planet as things change, as the planet ages considerably and begins to leave that habitable zone that I talked a little bit about last week. And we also might be able to think about kind of the processes that are happening deep in these really extreme environments, like at the ocean floor or inside volcanoes or other places where things are a bit more probably the way they were back when the Earth did start to have life for the first time, and aerobic life in particular. So this does kind of give us a snapshot and allow us to kind of think about how things would have evolved over time, and how all these things might spell changes on other planets and how they might allow us to pinpoint these other places that are kind of Earth-like and maybe habitable for us, or even just places where we might be able to contact alien life one day. Welcome back to the Wild Side. That was Mandy Moore with Song About Home. And the next thing I want to talk about is, in fact, a home. It's the home of birds and how the songs of birds can change as they move into new homes. And this is kind of an interesting study because it's by someone that I know from when I was still living in the U.S. And he's been working on chaffinches in the Canary Islands for about 15 years now. And this particular study is the culmination of something that he's been working on for a really long time. So it's quite nice to see this interesting result being published in a scientific journal and then also talked about a lot in the press as well, because it is quite an impressive study that he's done. So what he has looked at is how the songs of chaffinches have changed as they've moved from mainland Europe outwards into the Canary Islands. And the Canary Islands are the farthest point to which the birds have dispersed as they've colonized outward from the continent. And over the study period, the researcher, whose name is Rob Laughlin, he collected songs from 723 different males from 12 different populations, and that includes populations within Europe and also in the Canary Islands and then everywhere in between. So he's got this nice kind of gradient of songs where these birds are living. And he looked at all the subunits of the songs in order to do his analysis. And these subunits are called different things by different people, but he calls them syllables. And syllables kind of roughly equate to our words. So you've got a bunch of different syllables that work together to create the main song, just like our words create a sentence. And although different songs had different subunits, what he found was that the sequence of the syllables, or the syntax, if you like, of the songs was quite different depending on where the birds were. So basically they were using those same kind of building blocks of communication. They were using those same words, but they were rearranging them in a new way, and so their sentences sounded different. And this was actually something that you wouldn't normally probably hear if you were just to go out to these places and listen to the chaffinches singing. But if you put all of this into a computer and ask a computer program that's very sensitive to little tiny changes to kind of flag those up, then it does become much more apparent. And what Rob found was that the further away the birds were from the mainland, the less predictable their syntax was. So basically they were kind of, the degradation of the syntax seemed to have a relationship to the distance. And this loss of syntax structure was stepwise. So basically, the farther away the species got, the less and less predictable the syntax was. And at the farthest point away from the mainland, so Gran Canaria, the syntax was basically completely disappearing, and the birds basically have no rules at all for this particular aspect of communication. So all of this is interesting enough, perhaps, if you like birdsong anyway, but this raises a whole new question. It doesn't really stand alone, because it makes us wonder, why would all these changes occur? Why do you have these birds changing the way that they sing depending on where they're living? And this seems to be, well, there are lots of reasons, but one of the things seems to be that you've probably got fewer other bird species in the new habitat, and so there isn't as strong of a need for communicators to be really distinct in what they say in order to distinguish themselves from other birds in the environment. And if you've listened to the show before, you know I've talked about how birdsong can be used for lots of different purposes, and that includes males trying to get other males to stay out of their territory, and also males trying to attract females. So let's just stick with those two examples. And basically with both of those, you want to make sure that those other birds that you're singing to recognize you as a member of their species. They recognize you as a particular male, maybe even a particular male from a particular region. And you just want to make sure that there are enough distinguishing characteristics in your song that they can hear those things and know something about you. And if there aren't lots of other birds singing around in the environment, then it's not going to be as hard for you to distinguish yourself. There aren't going to be as many voices, there aren't going to be as many voices that are similar to yours, and so there's not a need to be quite as strict. And this sounds like a really convincing explanation for what's happening in the Canary Islands, except that on some of the islands where they do actually have more species, and so you would expect to have more structure, that's not the case, and also vice versa. So there has to be something else that's going on here as well. And Rob speculates that maybe this is related to the evolution of the genes that are associated with song learning. And we know that genes can be important in both song learning and song production, but he particularly thinks that song learning genes might be important here, because they are the genes that basically make sure that an organism is paying attention to the songs that it needs to listen to in order to learn. And we do know that if birds don't listen in quite the right way when they're young and when they're crystallizing their song, so that means they are kind of picking things up and trying them out and then narrowing in on exactly what they want to do. We know that if they don't do that very well, if something goes wrong, then they sound quite weird as they grow up, and they often can't communicate very effectively. So Rob thinks that something has happened to that learning process, and that it's probably genetic in nature, and that there's been some sort of a mutation or a change of some sort, regardless of how it arose, and then that was passed on from generation to generation, so that younger birds are less able to kind of pick up appropriate songs and replicate those as they get older, and so there's kind of this breakdown of communication. And he talks specifically about a process called cultural trap, and cultural trap happens in small populations, so for example those that you find on an island after a few birds have colonized, where you've got this evolutionary interaction between culture and genetic predisposition. So basically this would favor individuals that can recognize and learn a wider range of songs, and that sounds like quite a nice thing. But if you kind of turn that upside down, the flip side of it is, if you're recognizing and producing then a wider range of songs, that means you're probably producing more syntactical patterns, which would then mean that potentially you don't have that kind of strict adherence to a very particular syntax like what we see on the mainland. So this could be one of the things that's driving this change in song over time. Another thing that could be happening, and there's no need for these things to work in isolation, they might both be happening at the same time, is something called withdrawal of learning. And this might simply be that young birds in some environments have fewer tutors to learn from, and that's just because they have fewer birds overall. So again, when you've got a small population because it's just been an island's just been colonized, then there might be only 2 or 3 different males for a bird to learn from, and so they would then have to kind of improvise. If they don't, if they don't have someone there to kind of tell them all the different ways to sing, then if they do anything different that kind of sounds interesting, they might just use that because they think, oh hey, that sounded pretty good. It seemed to work in this interaction with this male over here or this female over here. And so they rely on their own improv rather than on replicating some other individual. Now, as always, I do like to kind of point out that this stuff is not just interesting in an ivory tower sort of setting as we're sitting up there theorizing about stuff. It does have some potential applications in real life. And you know, if you've listened before, again, I've talked a lot about birdsong, that studying song learning in birds is very important for studying language learning and understanding language learning in humans. And we do know that there are, across communicating animals, lots of similarities that we can use to kind of understand the different animals a bit better and kind of approach maybe medical treatments or kind of therapeutic treatments later on. So it might be helpful for us to kind of understand these potential gene-culture interactions in communication learning in order to figure out what has gone wrong in someone who's not able to speak very well and how we might be able to address that. So hopefully this might allow us to illuminate some particular genes or learning patterns or even the evolution of language over time in order to better understand how language works in humans or even in other species as well. That was Jackie Greene with When You're Walking Away, and you are listening to The Wild Side on Source FM. And today I'm talking a little bit about some of the most recent news in science, and I'm kind of all over the place. I've talked a little bit about atmospheric science, if you like, or ancient Earth history. I've also talked a little bit about termite gut microbes. Just before the break, I was discussing bird communication, and now I want to switch yet again to a completely new field and talk about the evolution of bipedal walking in humans. And I've discussed this a little bit in the past on the show, but I've talked about it more in kind of the terms of how did we evolve as mammals and kind of how bipedalism was an important part of that for some primates. I've also talked about kind of athletics and how bipedal motion is quite important, and a little bit about how actually a lot of human evolution is thought to potentially have been driven by the fact that we have the ability to run and to chase after prey and to tire them out, and that we maybe evolved hairlessness in conjunction with that ability to run so that we didn't overheat. Well, this is a completely different take on that, which is quite interesting because it's an issue that goes back quite a long way to 1925 to be exact. And basically researchers have finally seemingly proved a theory that was first raised back in the early 20th century. And of course you never can truly prove something in science, but they have come very darn close to supporting it quite well. And what they have found is seemingly a direct link between cranial anatomy and two-legged walking. And this was something that was first proposed by someone named Raymond Dart, who discovered our first bipedal human ancestor back in 1925. And this species was known as Australopithecus africanus, and his analysis of this species suggested that perhaps bipedalism was related to the position of something called the foramen magnum. And this is the hole in the base of our skull through which our spinal cord passes. And in humans, it's centrally positioned right under the brain case, and we have the head that sits on top of that. And that makes sense because here we are standing upright, and so of course you want the spine to be able to go up, all the way up your back and right into your head. But in chimps and other four-legged mammals, we tend to find that the spine is positioned more behind the head. And again, that makes sense because these animals are lowered on the ground most of the time, and so for them to be kind of in a normal position with the head not being stretched in any awkward way, then they need it to enter from the back. They need that spine to go towards the back of the skull so that they can continue kind of looking down and to the front as a dog or a cat might do. And in the current study, researchers wanted to investigate whether, in fact, there is this evolutionary link between the position of the foramen magnum and the type of walking that an organism does. They want to know, do these things really have a relationship, or did it just happen that that is the position of that particular structure in this particular ancestor of humans? And so what they did was they collected specimens from 71 different species from 3 different mammalian groups, so from marsupials and also rodents and primates, and they looked to see where the foramen magnum was on all of these species and what type of walking style the animals have. And what they found was that the forward positioned foramen magnum was found not just in humans, but also in other organisms that they called habitually bipedal. So things like kangaroos and kangaroo rats and jerboas. And the similar positioning to humans was observed in animals that had very close relatives that didn't have that same structure and didn't walk bipedally. So this seems to be something that evolved completely separately in each one of these animals. It wasn't like some early ancestor had it and then passed it on to all of us. Instead, each of these organisms kind of descended from another animal that had this kind of structure where it was more towards the back than right underneath. So this is quite an interesting thing where now we find that all these animals have converged on this same solution to needing to be upright and not have to have their head in an awkward position as they're moving around in order to allow the spine to hook up with the rest of the brain. So in addition to illuminating the morphological changes that were required to help us walk upright, this pattern can also be used to help us identify newly discovered specimens and try to figure out, are they more closely related to humans? Are they more closely related to apes? What type of locomotion do we think they had? So we can kind of use these little tiny morphological variations in order to get a better idea of how these animals lived and how they behaved. Now the very last study I want to speak about is quite a quick little thing actually, and I had it here to talk about last week and I ran out of time, so it's good that I have a chance to discuss it this time around. And you might have heard this in the press because it did receive quite a lot of coverage. And it's not a very good study, and that's why I wanted to mention it actually, is that sometimes journalists will find these studies that sound exciting but aren't very well done, and then they'll focus on it and kind of spread the word when maybe they shouldn't, because it should be kind of swept under the rug. And basically there were some researchers, actually from right here in the UK, who decided that aliens had been found in our atmosphere, and they were very excited that they finally had evidence of extraterrestrial organisms. And these researchers were from the universities of Sheffield and also Buckingham, and what they did was they sent a balloon into the lower stratosphere of the Earth in order to collect samples. And I believe the timing of this was kind of around the time that some meteors were coming through, and so they were hoping to collect samples that may have been permeated by the meteor, so maybe influenced by the meteor, and collecting things that potentially fell off of that meteor. And what they did was they found one of their samples contained something that they referred to as a diatom frustule. And this is the non-living outer shell of a type of algae species that's actually very common here on the Earth. You've probably already heard of diatoms. I may have mentioned them before on the show. They're found in all sorts of stuff, and they're really important in industry. So if you're a lady, you probably have these in your cosmetics. Everyone probably has these in their toothpaste. I mean, they're all over the place. And despite the fact that these types of algae are quite common on Earth, they're found in rivers and streams and oceans and all sorts of things, the researchers discussed the idea that maybe they're from the Earth, but they decided no, they're definitely from too far up in the atmosphere. They have to be from outer space. And their idea, because of the whole meteor thing, was that these diatoms were riding on the meteor and then they came off into the atmosphere. And this is in fact a possibility. I'm not saying that we have proof that that's not the case, but many astronomers have weighed in and said this is highly, highly unlikely. There are lots of reasons why you might have diatoms up in the atmosphere, and in fact you find that there are lots of little microorganisms that are up there. We know that bacteria and other things can float around and even reproduce while floating around way high up in the air, so there's no reason to think that maybe diatoms couldn't do a similar thing. And they can be ejected into the atmosphere by volcanic eruptions. They might also have been present right here on Earth, and they weren't in the atmosphere at all. It may have just been the case that diatoms got into the sampling equipment or got onto the equipment once it fell down from sampling up in the atmosphere. So it could have happened outside or even in the lab, that there was some contamination event and a diatom got in there somewhere. And most people seem to agree that both of these options are much more likely than the idea that there might be some extraterrestrial origins of this diatom bit. But people have long theorized that life on Earth emerged because there was some sort of organism out there that seeded us. So maybe these guys have actually found something, and I'm sure they have been quite attacked in the press, so I'm sure that they will want to try to replicate their research and find additional samples as well and prove that they were not a result of contamination. But just in case you did hear this covered and you thought maybe there was this good, highly supported evidence that there are aliens out there, don't get excited just yet. I think we definitely need a bit more proof of that before we can say anything conclusive. So that's it for me for this week. I'm going to end up with Milla Jovovich, the actress and also musician, singing the Alien song for those who listen. And this is from her folk album that she released several years ago. And I'll also add, just because I have the time, that if you like this song, she has lots of music that she tends to post on her website because she loves to dabble in music. And some of it's quite crazy, but some of it's also really interesting, and I am kind of a fan of her things. So hopefully you'll enjoy this. Hopefully you enjoyed the show and learned a lot, and I will talk to you next week.